Hello, everyone. Welcome back to the AAS YouTube channel. And this is part of the good stuff. This is part of the great stuff. This is the AAS Journal Author Series. And I am delighted to have Michaela Leung with us today. Hello, Michaela.
Hello.
Michaela, let's see. What's your geolocation? Where are you at?
I am in Riverside, California, where we are almost into early summer, to be honest with you. The summer starts very early here, so
Well, it's the
it's
Central
getting
Valley of
hot.
California. So, it
Yes.
moves up pretty good relative to the
Yes.
coast. Let's see. It is Monday, April 14th as we record this, and I'm in Phoenix. And we hit our first triple digit temperatures about 4 days ago. So now we're on a little bit of a cooling trend. We'll go back down into the 80s or 90s. So it's perfectly fine. And Michaela, what is your position here on Riverside?
I am a fifth year PhD candidate. I'm actually defending next week. So by the time this comes out, most likely, I will be degree in hand, I hope.
Well, let me
So
be the one of the first to congratulate Dr. Leung.
thank you.
Very
Thank
nice.
you.
Very nice. Very cool. That is awesome. And Michaela, on your thesis and otherwise, what do you like to do for research?
Yeah, so my main area of interest is thinking about biosignature candidates, really pursuing that, search for life beyond the Earth in kind of the exoplanetary context.
Cool.
By methods, I like primarily describe myself as like a photochemical and spectral modeler, which is like kind of a mouthful.
Okay.
But that's kind of the approach that I like to take is to think about, what happens to things produced by biology? Where do they go? And can we see them?
Awesome. Very cool. I predict a very awesome thesis in public PhD. Very good. And that is going to bring us to this very awesome APJ letters. It is open access. It's the open access era, people. You can go grab a copy for free. Go get one.
Yes, so
Examining,
this is
oops, let me go. Examining the potential for methyl halide accumulation and detectability in possible Hycean-type atmospheres. And Michaela, take us away.
Yes, this is actually, as you could probably guess, one of the chapters in my thesis. We worked on it, as part of my PhD, along with all of these other wonderful folks, Shami, Eddie Schwieterman, who's actually my PhD advisor, and then Daniel Ingerson and Janina Hansen as well. So kind of a good core team
Great,
chipping away at this problem from a bunch of different sides.
great shout out. Great shout out. Okay, let's go.
So basically, our approach to this particular project builds on some previous work that I did, which is also in my thesis. And we look at methyl bromide, so CH3BR, as a biosignature candidate for
Okay.
Earth-like planets. That's Leung et al. 2022 in AppJ, for the curious at home.
Very good. Very good.
And kind of what we found in that paper is that methyl bromide, like methyl chloride, which was previously established as a kind of a strong biosignature candidate, we find that methyl bromide shares many of the same traits and
Cool.
could then potentially be applicable as a biosignature. One sort of caution that we do find to that is that it is a little bit out of reach of what we could do with JWST and kind of pushing the limits of even like a next generation mid infrared telescope. And that's because the features that we looked at were kind of in this area of the mid infrared, where in particular, JWST, unfortunately, is just not super capable. And I think that fits with what other people have found
Okay.
as well. And so, kind of turning our gazes from a direct Earth analog, we started thinking about these temperate sub-Neptunes or the Hycean planets, as have kind of come out in the last couple of years, where
Uh-huh.
you have this hydrogen rich atmosphere over a surface liquid water ocean. And
Okay.
then you can get your standard temperature and pressure as we see here on the Earth, as well as a surface liquid water ocean. And the idea is perhaps with all of those features, we can call this a potentially habitable planet.
Cool.
And what is perhaps, equally exciting about these planets is that you can look at them with JWST. So kind of the perhaps type case for the Hycean planet is K218b, which has already had, I think, multiple JWST observations and was observed by Hubble and various other telescopes before that. And so we here are doing, yeah, exactly. We're doing some simulations for basically K218b to look at these methylated biosignature candidates. And I should, of course, note that in the JWST observations of K218b, there's a tentative, perhaps, detection of dimethyl sulfide. I think this is a little bit, they published this, then some other people came out and said, well, maybe not. And it's a little bit one of those back and forths. We probably need more data, kind of things to
Science at the
sort out.
bleeding edge. Science at the
Yeah,
bleeding
exactly.
edge.
Right, but this is a potential methylated gas biosignature that they're saying could potentially be detected on this potentially Hycean exoplanet. A lot of potentials in there, but that does sort of promote the idea that these are reasonable, places we can look for biosignatures, and kind of building on the previous work that I did looking at methyl bromide to look at methyl chloride, methyl bromide, and methyl iodide, which are all three the methyl halides, where you have the methyl, which is carbon and three hydrogens, and then halogen, and basically looking at that group of gases as potential biosignatures for the Hycean planets.
Okay.
Yeah.
And I will put a link in the description below the video to the other paper, the other chapter of your thesis. How about that? Okay.
Okay, perfect. Sounds good. I always feel funny plugging myself, but that is where this is
All
really
good.
a sequel paper to the other chapter.
So
like I said, in sort of methodology, I primarily work on photochemical and spectral models. I should say this Sayed 2024 paper that's coming up in the corner also looks at DMS on K218b and is part of the back and forth about it. And that's Shami is also one of the authors of this paper.
cool.
And we actually are using the code from that paper that does photochemistry for K218b. And that's the Vulcan model, which is kind of designed to do a variety of planetary types, but I think is often used for temperate sub-Neptunes, which is perfect since that's what we're looking at here. The previous work with this had looked at, other gases. I think we note sulfur, nitrogen, carbon, hydrogen, and oxygen is kind of what was previously in there. And then to look at these methyl halide gases, we're putting in a bunch more, reactions, gas fluxes, photodissociation data, things like that, just to really expand the capabilities of the model. And that expanded model is available on GitHub. It's down in the footnote of the paper. But you can check that out too if you're curious about looking more about what we did there.
Very cool. Nice GitHub link. We got 144 reactions in this one.
Yes.
Okay.
Yes. I feel like a lot of because I had to do a similar modification for my previous chapter on Earth-like planets in a different model, I feel like most of my PhD is just in adding reactions to models. But
yes.
That's how it gets done, right?
Absolutely.
So that's kind of the setup. And the other model that we use is the planetary spectrum generator, which I think is discussed perhaps on the next page, which is the open access suite out of NASA Goddard that does radiative transfer. So I think that is perhaps in the second. Yes, here we go. 2.2 planetary spectrum generator. Yep. And we're basically just using the model and the JWST templates that are within BSG, which we used for the previous sort of biosignature studies as well. So that's basically just the final piece of setup there is we're using Vulkan, BSG, and then we're actually running the models and looking at the results and specifically integrating, right, what photochemical results would look like in potential spectra. And kind of melding those two. So if we roll up a little bit, because I think we just rolled past figure one.
Let's figure one.
Yeah, we can roll back up here. Okay, perfect.
Okay, so we got it.
So I feel like this figure is primarily for the photochemists among us. But what we're looking at here in sub A is a comparison of the spectra of our two host stars. So I guess I should slightly back up and say that to simulate K218b, we're using the stellar spectrum of GJ436. And this is because my understanding is there's not a good short wavelength spectrum of K218b, which is actually or K218, this host star, which is, I think, a problem for I think a number of stars out there that people are interested in as exoplanet hosts is we just need more of that short wavelength data. And what we found in previous works for methyl halides and actually for methane is that for M dwarf host stars, you can see high accumulations of methyl halide gases. And so that's why we're particularly in this case interested, in addition to the observational advantages of the Hycean planets, that previous trend suggests that we could maybe have another reason to get high buildup of these gases that we could maybe detect on a Hycean planet.
Okay.
So that's why we're comparing the sun and GJ436 in the very top left plot there. And then below that, we have the photochemical cross section. Since we're using a photochemical model, so we're thinking about how the light interacts with the molecules and then breaks them up. And then there's kinetic chemistry from just the reactions shaking around, or the molecules just kind of shaking around in the atmosphere. And so that's kind of the key photochemical data. And what we get from putting all of that information into our code is what we have in subplot B, which is looking at the photolysis rates, which is basically how much these molecules are splitting, right? We have methyl chloride in green, methyl bromide in purple, and methyl iodine in orange.
Nice.
And you'll notice that methyl iodine has kind of the highest photolysis rate, followed by methyl bromide. That will be important later, but ended up in this plot just due to honestly the shape of the figure. That's where it is. Yeah, exactly. And then in the bottom row here in C and D, we're just looking at the atmospheric mixing ratios of these gases. And the reason why we have a whole bunch of lines for each gas is because we're actually looking at them across longitudes. And what we find is that the longitude like across the planet. So we actually did some two-dimensional simulations, basically to get a sense of, we're not completely doing like a 3D GCM type profile, but we are doing a little bit more than like the planet is one pixel. And this is also because I think we think a lot of these planets are probably tidally locked. And so then, thinking about where you are on the planet, there's some work by Howard Chen in, I want to say maybe like 2018 or so, that looks at the potential accumulation of gases like on the planet's terminator, right between the day and night sides, just because of like this circulation of the atmosphere. And so what we find here is that at least for the particular configuration that we tested, we're probably not seeing a significant day-night impact, except for methyl iodine. But the methyl iodine mixing ratio is so low that you're unlikely to be able to detect that in the atmosphere. So, like, yes, there is some longitudinal dependence, but it's probably not spectrally relevant. And this paper is primarily concerned with, like, could we see this right now? Like, could you quote JWST at a planet and try to look for methyl halides?
We have both.
So then there's a little bit of.
We have dashed lines and solid lines. What's the difference?
Yes. So the dashed lines are the 1D model and the solid lines are the 2D model by longitude. So what we're also showing here, and the 2D model is also by Shami Sai, and I think it's out in Sciadol 2024B last year. And we're showing that there's pretty good agreement between the model two, which is always nice, right?
I'm always impressed when somebody can cite a reference with a B on it. It's like, okay, you actually know that. Okay, and so the factor of 50 comes basically from this, right? Factor of 50 in the flux. Even then, higher.
So the factor of 50 comes in because we are exploring a variety of surface fluxes. So basically how these models work, right, is you set what's going on at the surface, and so you're just like fluxing stuff up into the atmosphere, and we explore a range of fluxes, and again, this is sort of like, this will be important later again, but the 50 times flux is about where we start to have a spectrally relevant level of these gases.
Got it.
So it's kind of just a contrast, both for like the exact conditions that we see on the Earth and the conditions where we think you might be able to start seeing something. That's kind of the two endpoints there. We do consider, and this will be in the next figure block thing, kind of a range of flux conditions. This the figures are just set up a little bit weird, honestly, because AppJ Letters has a figure limit, and so you kind of
Yes, you
just
do.
cram all of the
Oh,
figures
that's
into
how you did. That's how you got around this. I was gonna say.
I'm supposed to
Yes,
admit to that, but like that's like what happened here. That's why some of
yes.
the figures are you're like, this is just three figures in a trench coat, and like it is. That's what's going on
So
there.
should we go to figure two?
Yeah, let's jump to figure two.
Okay, let's take a look at this one.
Okay, perfect. So primarily it's actually the bottom. I was actually just reformatting this for my thesis, and I was like, that bottom actually maybe should have been at the top because that's our photochemical result there at the bottom. So what you're seeing on the x-axis is the flux of the methyl halides on the surface relative to Earth. So we're basically going from 1 times Earth to 1,000 times Earth, and that is a pretty broad range. But what we do see on the Earth is that there are local environments on the Earth that can produce like up to 1,000 times the globally averaged flux. So like you are looking at a global, perhaps like wetland environment, but you could somewhat reasonably, with what we know about biology on the Earth, reach that 1,000 times flux. That's why we're considering kind of that broad constraint.
Yeah.
And so we're basically showing that like for higher fluxes, we produce higher surface mixing ratios. And that we have our two
Yes.
star types. We have the sun, right? And we have that M2. And we're showing that for the M dwarf, there is enhancement of the buildup in the atmosphere, which, like I said, does match with the results that we've seen previously, that you get more accumulation for the M dwarf.
Cool. And what's the gold dash lines?
That's for our results for an Earth-like atmosphere. So that's what we find, the highest flux that we find in an Earth-like atmosphere. So for methyl chloride, you can see that we're exceeding that, especially in the M dwarf case.
Yeah.
Which again, like that's even more promising for potentially detecting these gases in the atmosphere.
Very nice. Cool. Very good. Okay.
So that's kind of, I guess, like the main bit of it that's like photochemical. This middle plot here, though, just real quick, in one of Shami's size 2024 papers, he shows the DMS one. He shows that if you have a bunch of DMS, it can also produce haze precursors. And what we're showing here is actually that we don't replicate that result for methyl chlorides.
Oh, yeah.
I suspect this has to do with the fact that methyl chlorides really want to reform. The reaction of reformation in the atmosphere is pretty favorable. And so I think instead of all of those loose methyl radicals forming into things like ethane or other haze precursors, they're just reforming, and you're just getting the methyl halides back in the atmosphere. But that's what's going on in this middle plot here. And that's kind of everything from the photochemical side. So basically, after that, we're taking our photochemical profiles and we're putting them into the spectral simulator and simulating for a K2-18b like planet. If you go right up to the little top panel here, so this is from the Hycean database. This is the input like base spectra of the gases as have been measured. And I'll just put a plug in here. You'll notice that this ends at like 5 microns. The gases probably have absorption short word of 5 microns. It just has not, or I guess short of 3. I guess I was looking at the thing slightly wrong there. Short word of 3. And there's probably absorption there. And half the world's observatory gonna look short word of 3. So there's a definite need to look more in that direction for sure.
Yes. Are these line intensities, are they all measured in the lab or some of them theoretical?
I believe these are all measured in the lab. And we actually have a grant funded to do more lab measurements in those very short wavelengths because there's so much interest, obviously, in maximizing the use for HWO. So we're hoping to conduct also some kind of really high resolution laboratory measurements to kind of build on this. But I think you can really easily see from looking at this figure, right? There's just nothing after 3.
There's a figure here. Yeah, exactly. Yeah. Okay. I'm with you. Very good.
Okay, awesome. So basically from there, we're taking these, input spectra from the gases themselves, and we're simulating them in the planetary conditions. And we simulate for 2 telescopes. So the first one is JWST. So I think that's our next figure. If you scroll down.
Your near spec. Yep.
Yep, yep. So we're doing two simulations here, one for near spec Prism, one for MIRI LRS. I think if you have looked at many spectra, you can look at this and see that what's happening in the MIRI wavelengths is the noise is just like we're being eaten alive by the noise, right? That's what
Yikes.
the error bars
Yeah.
are showing. And so even for our most optimistic flux cases where you have, 10 to 100 times what we see on the Earth, we're still kind of really struggling in the mid infrared. And what we found in my previous work is that 10 micron feature is like was the most favorable. But when you're considering the JWST constraints, the 10 microns starts to look not so good because of all of the noise there. And if you look at the bottom set of subpanels here, we're
Mhm.
looking at the number of transits
Okay.
necessary to detect these gas features. And you'll notice that the scales are not the same. For our near
Like,
spec prism, we're looking at like up to 15. And for the MIRI LRS, that actually goes to 100. And so basically to see any methyl halide feature, you need a minimum of like 25 transits. Um, but if you want to have, higher confidence, you're looking at potentially, 70 to 100 transits, which is a huge time investment.
seems like a lot.
Um, yes, I think it's very unlikely that any single person or target will ever see that much time with JWST. Versus in our near spec prism range, we're seeing between 5 to, I believe, 14 transits, which
Okay.
is
Okay.
still, it's not nothing, but I think you're much more likely to be able to argue for 5 to 14 transits with JWST versus is a number that hits 100,
And
right?
even if you get 5 or 15, right, it's a start, right?
Right,
You got to
right,
start.
totally. I mean, you could, and I think a lot of what we envision with kind of this group of methylated gases in general, anyway, is like looking because they do seem to require a little bit more time investment, looking for kind of the classic, if you will, like first line biosignatures like oxygen, methane, things like that. And then once we kind of have something that's really promising, going back and looking for this specific group of gases. Because they have very low false positive potential. So that's an issue with things like oxygen, methane, things like that. Is there's all of these other ways that we've thought of that you can get kind of planetary scale amounts of oxygen or methane abiotically. And the processes to produce methyl halides abiotically are really, really limited. And so it's very unlikely, in my opinion, that you could get that at a global scale. Of course, who knows what's out there, but it does seem like if we have not been able to convincingly figure out what that would look like, hopefully this is a clear, it'd be perhaps more straightforward if you detected methyl halides to interpret them as biological versus something like methane, for example, right, that could come from volcanism or serpentinization or like all of this other stuff.
Right. Cool. Very good. So this was for JWST. Okay.
Yes.
So the next figure is probably going to be for ow.
Yes. So our other telescope simulation here is for life. And this is done specifically by Daniel Angerhausen and Janina Hansen, who are on the life team at ETH. And so basically, in contrast to looking in transit with JWST, we're looking in emission. And so
Yes.
that section right there, A, is going to be just the raw kind of emission spectrum just from the radiative transfer model. And then we basically run that through LifeSim, which is a code developed by the Life team to constrain the noise for the life mission, which is what you see in subpanel C. I'll come back to B in 1 second.
Uh-huh.
But you're looking at 24 hours of observation,
Nice.
which is not too significant of a time investment. And in the bottom plot, you can see that for especially our 10 or our 20 to 100 levels, we're looking at really potentially high significance for feature detection.
Signal.
And we have another paper, which is Angerhausen et al. 2024, where we look at a similar result for the Earth-like planets. And I will tell you, this really kind of blows them out of the water in terms of how easy it would be to detect these gases.
Cool.
And my understanding is that there's like an initial survey phase for the Life mission in which 24 hours of data just would be captured. And so it sounds like perhaps there would not even need to be a dedicated
Uh-huh.
observing campaign on this target. That data would just come as part of that initial phase. And additionally, what we're seeing in section B is the expected yield for the Life telescope, which I think is maybe part of LifeSim or maybe an ancillary software, which is suggesting that we could see in the 3 to 5 parsecs region, anywhere from like 14 to 30 almost, depending on how exactly you define your like Hycean planet, whether it's a super Earth or a sub-Neptune, or, it's all a little bit, it's kind of a little bit unclear, I think, with these Hycean planets exactly where
Yep.
they would fall in that size distribution, or maybe they would cover the whole size distribution. So it does suggest that, there are a good number of these targets that are potentially detectable with really minimal time observation. So this is kind of, part of a contribution to the science case for the Life mission kind of development team as well.
So that is it's not a particularly small yield. That's good. There we go.
Yeah, yeah.
Okay. Oops, wrong way. Okay, very cool. Awesome.
So that is, I think, pretty much the meat of it. We do discuss here in the discussion a little bit, like some of the small abiotic sources. There's an extensive discussion about that in my other paper,
Yeah.
which is really long. So this is the short version.
Oh, okay.
But we're basically kind of summarizing, like we could do this with JWST. There's kind of a plug for these measurements that I was talking about earlier with HWO, since we're running to really short wavelengths. And kind of, I think, feel like the next phase of biosignature science is really all shorter than 2 microns.
Sure, yeah.
At least that we know of, since the Life mission is like pre-selection. So if that were selected, obviously that's a potential mid-infrared target.
That's an issue.
Yeah, right.
It's
So
never
really, this is kind of our argument for doing more work on these gases, and thinking about like I said, we mentioned a little bit some of the abiotic sources, and discuss a little
yes.
bit like our
Nice.
sources, limitations, all that good stuff. But really, I think this is kind of a majority, it's a kind of just a proof of concept of like we could see methyl halides somewhat reasonably with
Uh-huh.
JWST, and kind of just putting that out there to the
Mhm.
community, especially if we come back and see some of these other targets have the kind of same promising nature with these maybe frontline kind of biosignature candidates.
Yes. Very cool. Awesome. Very nice. And just to round off, we have a little appendix here on
Yes.
what our boundary conditions are.
Yes, this is mostly for replication. So
Mm-hmm.
if you want to get out
Good
your
thing.
Vulkan model at home and recreate this, or your own other photochemical model, right, for comparison.
Mm,
And these are all of our softwares,
very nice.
and our funding and all of that stuff.
Tom Wag, I did a video with Tom just a few weeks ago. Very good.
Oh yeah, he's got this software
Yeah.
for like putting all of your citations in, which is really
Yeah.
useful because you're like, I don't know whose NumPy paper this is, right?
Very cool. Michaela, I want to thank you so much for walking us through your very awesome article. Very nice.
Thank you. Yeah, I am glad we got this out for sure.
Sure. Sounds like I'm going to be putting a few links to your papers in that description below the video. So you touched on it a little bit, a couple of times actually. And so let me go ahead and push on it a little bit and ask, where do you think we go from here, given the published article? What are sort of the most immediate next steps? Is it line lists? Is it opacities, measured opacities in the lab? Is it additional models? Just where do you think we go for next steps?
Yeah, so I think there's like a couple of directions. So there's, like I talked about a few times when I was going through the paper, the short wavelength
Yes.
line lists to measure, to understand, like, is this something we could do with HWO?
Right.
In addition to that, there are a lot of other like gases kind of that are also produced by the methylation process that could also be considered as biosignature candidates. And we've looked at kind of Earth-like planets and these Hycean planets, and there are, of course, other, like there's the Archean Earth, right? There's other potential planetary configurations that we could consider. So those are kind of the three directions I would say: is the short wavelength data, the other gases, and potentially other planetary types as well.
Well, I'm predicting a fairly fruitful postdoc, Dr. Leung.
Thank you.
Very cool. Well, it'll be really fun to see this develop over the next couple of years as we start hunting these down and looking for biosignatures on this kind of stuff. So it's very, very nice. It'll be exciting to see it develop. Very cool. And that will do, everyone. And I hope this made your astronomy day, your habitable astronomy day, just a little bit better. And we will see you on the next one. Bye-bye.